The today's topic is enzymes. Enzymes are substances that act as a catalyst in living organisms. Therefore, they are also called biological catalysts, regulating the rate at which chemical reactions proceed without itself being altered in the process. They themselves do not take part in a chemical reaction, however, they only catalyze the chemical reaction. Catalyze means that if a reaction needs 2 hours to proceed, with the use of enzymes, the time period can be simply half an hour. The biological processes that occurs within all living organisms are chemical reactions. All these chemical reactions are mostly regulated by the use of enzymes. Without enzymes, many of these reactions would not take place at a perceptible rate. We cannot wait for a long time for a reaction to complete. However, we can use enzymes so that a reaction can be proceeded in a much better and mm, in a less time. Now, enzymes catalyze all types of cell metabolism. They are involved in the digestion of food. They can break down the large proteins, carbohydrates, nutrients, fats, along with the conservation and the transformation of the chemical energy. And they are also involved in the construction of cellular macromolecules from smaller precursors. Okay, there are many diseases that are associated with the decreased level of enzymes, with the deficiency of a particular enzymes, and they are albinism and phenylketonuria. These two are mainly some inherited diseases that are result from the deficiency of a particular type of enzymes. These enzymes also have valuable industrial and medical applications. They are mostly used in the process of fermentation at industrial level. For example, they are used in the fermentation of wine, leavening of bread, curdling of cheese, and the brewing of beer. But not until the 19th century were these reactions understood to be the result of the catalytic activity of the enzymes. Although these reactions were taken place before 19th century, but till then they were not it was not understood that these are taking place with the help of enzymes or they are catalyzed by the enzymatic activity. Now, most of them have industrial importance. These enzymes are also known to kill the disease-causing microorganisms. They are also known to promote the wound healing, and they are also used in the diagnosis of certain diseases. Chemical nature. All enzymes were thought to be proteins at once, but after 1980s, it was known that there is an enzyme that is ribozyme, which has nucleic acid nature. So it was demonstrated that not all enzymes are protein in nature. However, most of the enzymes are protein in nature, but some can also have other natures as well. For example, ribozyme has the nucleic acid nature. Okay. Now, a large protein enzyme molecule is composed of one or more amino acid chain called polypeptide. As it is known that most of the enzymes are protein in nature, therefore there are certain therefore they are made up of amino acids. These amino acids are linked together by peptide bonds, or we can say that these are the polypeptide chains that are present in the enzyme. Now, as being protein in nature, every protein has its own specific structure. So, it is the characteristic folding patterns of the protein structure which is essential to the enzyme specificity. For example, an enzyme that is functional in the quaternary structure, when it is folded into the, for example, secondary structure, it will not function until uh, it takes the shape of the quaternary structure. Therefore, the enzyme has specificity for the protein structure. Now, being protein in nature, it can be subjected to various changes or fluctuations. For example, if you change the temperature or the pH of the protein or the enzyme, it may lose the enzymatic ability as the protein may denature in that case. Now, denaturation is sometimes reversible, but it can not always be reversible. These enzymes can be bound to additional chemical components that are called the cofactors. These cofactor is a direct participant in the catalytic activity and thus is required for enzymatic activity. Like some enzymes cannot be functional until they have this cofactor attached with them. This cofactor may either be tightly bound to the enzyme or may be loosely bound to the enzyme. Now, if it is tightly bound to the enzyme, the cofactor is referred to as a prosthetic group. Okay, then is the nomenclature of the enzyme. The nomenclature of enzyme is simply an enzyme will interact with only one type of substance or group of substances called the substrate. We always have an enzyme that is bounded to a substrate, and that is called enzyme substrate complex. Now, because of the specificity that every chemical reaction can be preceded by a specific type of enzyme, because of this specificity, enzymes often have been named by adding the suffix ase to the substrate's name. For example, the enzyme that is catalyzing the breakdown of the urea, it is named as urease. But we can say that not all enzymes can be named in this manner by this nomenclature. So we have developed several classes of the enzymes. Six principal categories are involved that are based on the chemical reaction it is proceeding. First one is oxidoreductases. Oxidoreductases are those enzymes that are involved in the redox reaction, that is involved in the oxidation reduction reaction, that is involved in the loss and gain of electron. Now it says which are involved in the electron transfer. Next one is transferases. Transferases are those enzymes which transfer a chemical group from one substance to another. So they are involved in the transferring of a chemical group from one substance to another. Next is hydrolases. Hydrolases is which cleaves the substrate by the uptake of a water molecule. They are involved in the hydrolysis reaction. Lyases, which form double bonds by adding or the removal of a chemical group. Isomerases, which transfer a group within a molecule to form an isomer. They are involved in the formation of the isomers. And last one is ligases. Ligases or synthases are those enzyme class which couple the formation of various chemical bonds to the breakdown of a pyrophosphate bond in adenosine triphosphate or a similar nucleotide. We can simply say that ligases are those enzymes that are that are involved in the formation of various chemical bonds. So these are the six principal categories of the enzymes based on the reaction type it is proceeding. Now, mechanism of enzyme action. This slide is a bit dumped with the text. However, I'll explain it. Now, mechanism of enzyme action is that these enzymes are responsible for the lowering the activation energy. It is involved in lowering the activation energy. Plus, whenever an enzyme substrate complex is being formed, there forms a transition state. They react with the substrate to form an intermediate complex, a transition state that requires less energy for the reaction to proceed. The unstable intermediate compound quickly breaks down to form reaction products, and the unchanged enzyme is free to react with other substrate molecules. Only a certain region of the enzyme called the active site binds to the substrate. Now we have an enzyme, we have a substrate. That enzyme has an active site on it. The substrate will bind to that active site. When it is bind, a transition state is formed by the enzyme. When this transition state is basically involved in lowering the activation energy for the substrates to form product. When the activation energy is lowered, it directly forms the product, and thus that enzyme is set free to bind to other substrates and proceed those chemical reactions. So the mechanism of enzyme action is that it lowers the activation energy required for a chemical reaction to proceed by forming a transition state. After the formation of this transition state, directly products are formed, and the enzyme is then set free to bind to other substrates and form their product. Okay, the last one heading is the enzyme inhibition. Okay, now what is enzyme inhibition? We have an active site at the substrate, at the enzyme. That active site is used for the binding of the substrate. Now in this case, inhibitors are present that come and bind to the active site of the enzyme, thus inhibiting that active site to bind with the substrate. Clear? We have enzyme inhibitions There are inhibitors. One method to accomplish this is to almost permanently bind to an enzyme. These type of inhibitors are called irreversible. However, other chemicals can transiently bind to an enzyme. These are called reversible. Reversible inhibitors either bind to an active site or to another site on the enzyme. Okay, we have inhibitors. These inhibitors bind to the active site of the enzyme, therefore blocking the site for the substrate to be bound. When the substrate cannot bound, the reaction cannot proceed, and that is called enzyme inhibition. Now, this can be of two types. Either it will be irreversible or it can be reversible. In the case of irreversible, the inhibitors will then detach from that enzyme. Sorry. In case of irreversible, there is no way out. That is a permanent blockage for the substrate. And in case of reversible inhibition, and the inhibitors will then detach, and then the substrate can bind and the reaction can proceed. Okay, now we have competitive inhibitors. These competitive inhibitors will proceed. Here it is written, competitive inhibitors will bind to the active site and non-competitive inhibitors will bind to other sites on the enzymes. Now, competitive inhibitors will bind to the active sites. They compete with the substrate at the active site and therefore increases the Km. This Km is basically the affinity of the enzyme. However, Vmax is unchanged because with enough substrate concentration, the reaction can still complete. Vmax is the condition in which the enzyme is fully saturated by the substrate. Okay. Now, when we have enough substrate in that chemical reaction, when we have enough substrate, the reaction can still proceed. And therefore, the Vmax or the enzyme saturation by the substrate is unchanged. However, Km increases, that the affinity for the substrate by the enzyme increases. And in competitive inhibitors, the inhibitors are bind to the active site. Same, we have non-competitive inhibitors. Now, in case of non-competitive inhibition, what basically happens is that it binds to other site on the enzyme other than the active site. It binds on the sites of enzyme other than the active site. The active site is still present for the binding of the substrate, and therefore the reactions will proceed. In this case, the Vmax decreases. That means the saturation of the enzyme substrate decreases, and whereas Km is unchanged, the affinity for the substrate to bind with the enzyme remains unchanged. This is a simple competitive and non-competitive inhibitors difference. Where this attacks, it acts on the active side, and the non-competitive competitive inhibitors act on the alternative side. Km increases and unchanges, and Vmax unchanges and decreases. Okay. Next one is phosphorylation. Now, what does basically phosphorylation mean? It is also another mechanism by which enzymes can be inhibited. What basically happens is that proteins, okay, we'll read it from here first. This typically occurs through the action of kinase enzyme, which can either inhibit or activate an enzyme depending on the situation. The kinase enzyme cleaves off a phosphate group from ATP and binds it to the enzyme. In situations where this results in an increase in enzyme activity, it creates a cascade reaction, allowing a large response to be generated from a small stimulus. Okay, what basically happens is that kinase enzyme is responsible for this inhibition of enzymatic activity. What kinase enzyme basically do, it basically cleaves off a phosphate group from the ATP, converting it into adenosine diphosphate and a phosphate group. This phosphate group goes and binds to the enzyme, therefore blocking the substrate to bind with the enzyme. The reaction is then stopped. In this case, it also creates a cascade reaction. A cascade reaction is that reaction in which allowing a large response to be generated from a small stimulus. Whenever it receives a small stimulus, a small response, a small query, it will respond in a better, larger way than others. So it is basically, it will create a mismanagement type case in this phosphoryl, in this the enzymatic activity. Last one is zymogens. Zymogens can also be, enzymes can also be secreted in an inactive state, which are called zymogens. Zymogens are a useful mechanism which allows enzymes to be safely transported to different locations without the enzyme becoming active and performing its function along the way. Now, sometimes we do not require the function of the enzyme, however, we want it to be at its location whenever it is needed. So, in that case, these enzymes are secreted in the inactive state. When it is secreted in the inactive state, it is called zymogen. It is a very useful mechanism in which they can be transported very easily to different locations in the inactive state. Inactive state, so that when it is needed, they can perform function. They remain inactive due to an addition of amino acid in the protein. Therefore, to activate a zymogen, another enzyme must leave off these additional amino acids. For example, chymotrypsinogen is synthesized by the pancreas, yet is inactivated and cannot function. Upon traveling to the intestines, other enzymes leave off the additional amino acids to produce the activated form chymotrypsin. What is what happens is that these enzymes are inactive. Why these are inactive? Because they have this, they have been added with an amino acid. So to make them functional or to make them in the active form, this amino acid should be cleaved off from this enzyme. For example, an example is given that chymotrypsinogen that is synthesized in the pancreas, but it is in the inactive state. When it is traveled down to the intestine, trypsin enzyme breaks down this additional amino acid and it produces the activated form, that is chymotrypsin. So this was all about enzyme, its activity, its inhibition, and its nomenclature. About the active site of enzyme, we have read that enzymes have active site to which the substrates bind. For example, this is an enzyme. Now this enzyme has, this is the active site of the enzyme. For example, we have two types of substrates. First one is this and the second one is this. We have two different types of substrates. This is the substrate 1 and this is the substrate number 2. Which enzyme will which substrate will bind to the active site of enzyme, that which can fix in this place. That is the substrate number 1, which will bind to the active site of the enzyme. We have a lock and key model. We have a lock and key model to explain this. That means we have different keys and we have one lock. The only key that will open this lock will be the one that will fix into that place. So the substrate that will fix into the active site of the enzyme will bind to the enzyme and form enzyme substrate complex. This is now an, because it has an enzyme which is binded to the substrate, that is an now an enzyme substrate complex. This was all about the enzymes, its reactions, the nomenclature, the inhibitions, and the all about the enzymes. If there is still remains any query, you are most welcome to leave it in the comment. Thank you.